first. So first of all, thank you for joining us. Don't forget, we have a very exciting talk happening next week from Amita Gurur of the Zeiss Research Group. She's going to be talking about basically some new things coming out at Zeiss. And as always, we have the events calendar. Hard to believe we're halfway through April already, but here is kind of an overview for the rest of the month. You can always find that on our website. The link is at the top there. We have other user groups if you are at all interested. We have 3 other user groups. We have the AIM user group, the lattice light sheet, and the expansion microscopy. Again, if you'd like to join any of those, just go to our webpage and take a look at them. Very exciting news. We will be having our yearly community congress in Montreal in October. Registration will be opening soon, hopefully next week. And we hope to see you there. There's also, the week before that, a course being put on by Global Bioimaging and the Canadian Network of Scientific Platforms about core facility management. So that would be, if you're interested in that kind of thing, it would be, you could come to there and just stay for the rest the following week. All right. This is just a little bit about us. BINA is a group meant to build community and disseminate technology, and we're heavily focused on community building and having everybody learn new technologies. Membership is free. You do not have to be a member to join any of these things, but we always appreciate it. Right. This is possible by a number of sponsors, including the Chan Zuckerberg Institute. These are some of our platinum sponsors this year, and they make all of this possible. All right. Just a quick reminder, this user group aims for an inclusive environment where everyone is welcome, and we expect that all participants will honor that goal. And now I'm going to do a quick round of introductions. We have Ethan, who is kind of one of the leaders of this user group, and Daniel. And then we have a number of other folks who are heavily involved in this. I know Thomas is on here. I don't know if anyone else is. And then, of course, there is the BINA program team, me, Alex Cotton, and Nikki Biley. All right. There is a document that we have shared that has all of the resources and directories, and we'll keep adding to that as we go on. And now here's our brief agenda for today. And then I'm going to pass it over to Ethan to introduce our speaker.
Hi, everyone. So I'm really glad to see some participants who have come back for several sessions, and this really is about community, and I think that we're having some success in that. So it's really great to see some repeat faces and some new faces as well. I do want to thank Alex for that introduction, and also mention once more the open notes, which is for our use as a group to use in ways that we want to add notes, things that we think are important findings coming out of today's meetings, and including the discussion that we'll have at the end. We also have a few other documents that are linked at the top of that, which include the voluntary directory. Again, this is this voluntary directory, I've said it before, I'll say it again, it's not a mailing list. It's really just to hopefully connect users in this community to others, users with experience and expertise in areas of interest for the STED community. So if you haven't, we really encourage you to add your name and your contact information and any of your skills to that document. We also have a running list where we try to uh, collate the antibodies and dyes that have been people have had success with in their experiments for STED microscopy. So if you do have experience, please I encourage you, and also through your discussions that we'll have later in the meeting, please encourage those who share their skills to also add those skills in a tangible place in that document. Um, and so I think that's what I'll mention. We're really delighted today to have a joint presentation from Matthias and Jeffrey, who are both going to talk to us about insights across multiple imaging platforms. So, without further ado, I'll hand it over to them. I will say one more thing, actually, is after their presentation, we will have Q&A, and we will have also open a few discussion rooms. One of those discussion rooms will be continuing up with the speakers. Another discussion room will be actually focused on selecting some presenters for the GBI Cross BINA 2025 conference that Alex mentioned. So you can actually, voice and offer suggestions about who you might be think might be a great speaker at that really exciting event that's going to happen later this year. And then we'll also have a general discussion room. So we hope you'll stick around for that after the presentation. So now, without further ado, Matthias and Jeffrey, I hand it over to you.
Okay, thank you very much. First, thank you very much for this opportunity, and also thank you for Dr. Guang Gao at UBC who connected us and gave us this opportunity to give this presentation. So as Ethan mentioned, we are going to talk about not only about STED, just about the combination of this technique called the magnified analysis of the proteome, which is a sort of expansion microscopy that we applied across different imaging platforms to try to enhance the capabilities to visualize synaptic proteins. So just to begin with, at the lab, I am studying mainly synaptic proteins and trying to visualize them in the tissue. However, as you can see in this cartoon, it is very crowded in protein elements. So when you want to label specific proteins in this structure, it becomes very complicated to visualize them at the individual synapse level, especially when you are in this region of the brain that is called the neuropil, where you have a very high density of synapses. For a very, very long time, the gold standard to study these was the electron microscopy using antibodies coupled with gold particles. And so you would be able to just look at a region with very dense in electron, like this region that would be the synapses. However, limitation be the volume that you can acquire, which is quite small. It's very labor intensive. And also it's very hard to do any sort of multiplexing because you can only use one type of antibody coupled with gold. And if you try to just use standard light microscope, even sometimes super resolution microscope like the STED, for the standard confocal microscope, it's limited in resolution, which is not the case for STED. But another big limitation is the antigen accessibility. So again, with this very high dense region in protein, it's sometimes very hard to label them because it's not very accessible. And so with this in mind, in the last 8 years now, in the last 8 years, you have this technique called expansion microscopy, and among them magnified analysis of the proteome, which have been developed. And so just to give you an overview about this, the idea is to make a tissue physically bigger. So you add a physical magnification on top of the optical magnification. But it's not just about making tissue physically bigger. It's also removing a lot of cellular components that are usually generating background when you try to do standard IHC. And so by removing all of these lipids, sugar, you get more antigen accessibility for your proteins of interest. And lastly, one of the main advantages, because it's at the end filled with water without any lipids in the tissue, it becomes fully transparent. And so as you, most of you know, when we try to excite fluorophore, if you have a tissue that is quite thick and with a lot of lipids, with a lot of cellular components, you have like a scattering of the light within the tissue. And so you may lose signal of the fluorophore, but also excitation of the fluorophore deep in the tissue, which is not the case when the tissue is completely transparent with this technique. About more detail on these techniques specifically, the way it works is that you are going to use these molecules called acrylamide. So you have different type of acrylamide that you are going to immerse your sample in. And so what you want to do is this step called the gelation. So you're going to create this mesh, this network of acrylamide that are going to covalently react with your proteins and to some extent also with your RNA. And so then you're going to have this very dense, very complex gel that is going to be created with the proteins and DNA anchored to this network, to this matrix. And then what you want to do to expand it is to wash it with detergent, so mainly SDS plus salt, very high concentration of salt and SDS to remove most of the lipids in your tissue, as well as all of these bounds, these non-covalent bounds between your proteins. So you denature your protein, which can be one limitation we can talk about later. It's because you're going to denature your protein. Usually standard antibodies working with this technique, but if for some reason antibodies that you are usually using are recognizing 3D pattern of your protein, you may not be able to use it in that context because the protein is going to be fully denatured after the denaturation step, so this expansion step. And then by just washing it with first with PBS and Triton, and then ultimately switching to water or very diluted PBS, very diluted solid solution, you will just have like an osmosis. So the tissue, the gel, this hybrid between the tissue and the gel that is going to be inflated with having the water going inside the gel and making it bigger. In standard, it's about 4 times expansion. For this specific procedure, people have succeeded in the past of doing multiple rounds of embedding of the gel and denaturation, resulting to an expansion factor of around 10, if not, I think maybe one group did even 100. So you can really make tissue bigger. And so by pulling all of these components apart, you are really adding much more magnification to then the imaging when you're going to use your microscope at the end. So we published a paper about this, about adapting a little bit the initial protocol that was published of the magnified analysis of the proteome. In our case, studying specifically synaptic protein in different samples and also across different cell type. And in this paper, we also looked at how different imaging system performs when you combine magnified analysis of the proteome with these imaging systems. So we are not going to go over the entire paper today. We are first going to see how this technique improves visualization and quantification of synaptic proteins, because this is what we are studying. And then we are going to look into which step of this procedure is actually enabling this improvement in visualization in spatial resolution. And then I will let Jeff talking about the combination between this technique and different imaging system, how these couples. And so just for you to know, we are going to look at the brain and at this region called the hippocampus, specifically the subregion called the CA1, in this region, very dense into synaptic proteins, just to show how this technique really improved the visualization of synaptic proteins, because this region called stratum radiatum, it's where most of the dendrites of this region are, and is very, very dense into synapses, excitatory synapses. So also, just to give you a bit of highlight for people who are knowing a little bit about synapses, we are going to look at these different proteins. So one called PEN-MAGUC, it's a scaffolding protein on the postsynaptic side. ULX, which is an active zone marker on the presynaptic side. And then glutamatergic receptors, as well as voltage-gated calcium channel. So we chose all of these proteins because they are very close in space, very tightly. They should co-localize in space, and like with the best imaging system, you should even be able to tell them apart, even if they are very, very close in space. And so by using these proteins, we can really define synapses by having, for example, the overlapping of receptors with the MAGUC protein, or the alignment between the MAGUC protein and the ULX protein. So first, about just general improvement, the way we characterized it is by processing brain in two fashion. First, by just doing standard IHC and having the tissue being fixed with regular PFA and doing standard procedure to label protein on this tissue. So as you may see here, as we are talking, we are in the CA1 and specifically in the stratum radiatum, where it's very dense in synapses. And so maybe first what you can see is, like, for some of these markers, especially, for example, calcium channel 2.1 and ULX, the signal is pretty homogeneous across the entire tile here. And especially in this region where you should have only the nucleus and not synapses, you still have a lot of signal for these two proteins. So which shows that you have a lot of background signal that is non-specific. But when you process a similar brain with the MAP procedure, and you do then again the standard IHC procedure, you can see much more contrast between what would be the background versus what would be the specific signal. And especially when we look back at these two proteins, CAV2.1 and ULX, this time you can see that where the nucleus stands, you don't have any signal, which is expected, and you have very strong signal only in the stratum radiatum, which is this part with this high abundance in synapses. Then when we zoom in, which is interesting because we want to look at the individual synapse level, and we try to locate all of these synaptic puncta, we can see that only in the tissue that has been processed with this method called MAP, you have an alignment, a co-localization of this synaptic protein. Especially what we can appreciate here is ULX and MAGUC. We can see that the signal is really aligning for the MAP processed tissue, while for the tissue processed with standard procedure, we cannot see any of this alignment. All of these bright puncta might not be specific to these different proteins. And so we really don't have a good resolution of this synaptic protein. And then we try to quantify it. So the way we quantify it is by applying a mask to localize only the bright signal in our images, and the goal is to find how much of this puncta are colocalizing, because all of these should be located in the same space. And then using the same images, but like by rotating two of the channel to try to randomize the signal and see if we get the same amount of colocalization or if we lose this high amount of colocalization, to try to control just the random colocalization of our imaging. So what we can observe is only in the case when we process the tissue with MAP, we have a significant improvement of the amount of colocalization of our synaptic protein versus when the two channels are flipped. While when we do these for the IHC images, the standard IHC, there is no significant difference between the flipped images and the normal image, showing that you just don't have an improvement into the visualization of this protein, but also an improvement in how you can quantify them and how reliable is the quantification at the individual synapse level. We also did this for the glutamatergic synapses, the glutamatergic receptors, which are very important component in the excitatory synapses, and the results are very similar. So, also something that you mentioned also initially is like you like sharing the antibodies that are working with the different experiments you are doing. So in the paper that we published, we provide a free access to a list of, I think it's around 40 different antibodies that are working with this procedure that we can definitely share for everyone who would be interested into trying to replicate these experiments. And so now about what actually provide this improvement into visualization. So just to give you a little reminder, there are two main steps into this procedure. The first one is doing the gelation and then the denaturation, so removing all of the lipids and cellular components. And then there is the physical expansion. So we wanted to know if the main improvement that we saw here is due solely to the denaturation, so the removal of all of these lipids, all of these cellular components, or if it's the actual mechanical expansion that provide this improvement into this synaptic protein visualization. So the way we did it is by processing a tissue with MAP and then imaging it after the IHC when the tissue is still partially expanded. So as I was telling you, this expansion is due to like osmosis exchange. And if you keep the tissue in a quite salty solution like PBS, standard PBS, you are going to keep the tissue not fully expanded. So it's still going to be a bit bigger than the original size, but it's still going to be very limitedly expanded. So here we would be like around 1.4 expanded. And so what we can observe here is even though it's not fully expanded yet, you can still visualize the synaptic protein at the individual puncta. For example, if we look again at ULX and Penmagook, they are aligning, and then you can see some bright signal for CAV 2.1 that is going to locate somewhere between Penmagook and ULX. So what we can see here is like really it's mainly the denaturation, mainly the removal of the lipid and the increase in antigen accessibility that really provide this improvement in protein visualization. And then what we did is expanding exactly the same tissue and then image with the same condition this tissue and see what signal we observe. And so when it's fully expanded, but we use the same imaging settings as for the 1.4x expanded, we don't see any signal. What happened is by physically expanding your tissue, you are going to pull apart your fluorophore, which ultimately when you're going to try to excite them is going to dilute even more the signal. So that's a consideration I wanted to talk about, is by doing this physical expansion, even though you're going to gain more resolution, visualization, you're also going to dilute your signal a lot. And so this is a consideration that you have to have in mind. You need to have either a very sensitive imaging system in order to really get the signal coming from this individual puncta or your signal, or you should use much more antibodies that you would use for standard procedure, for standard IHC procedure. And so, but then when we adjust to the optimal setting, the imaging, you can really get a very high contrast image. And what you can start to see here is more than the alignment between the synaptic component, you can see within the synaptic component brighter signal, which might be what we called in our field nanocluster, nanodomain, which within the synapse are like higher density region in protein. Here, for example, you can see that ULX is not just a homogeneous line in front of MAGOOC, but is really high density puncta that are going to align in front of one MAGOOC. So when you expand the tissue and you do some imaging, you can get even more resolution, even more details into your signal. So now I'm going to, we also did this with the glutamatergic receptors. And so now I'm going to let Jeff talking about mapping combination with the different imaging system.
Okay, thanks, Matthias. So I guess we're very fortunate at UBC. We have a lot of different microscopes we can potentially take our expanded samples to. The LSM 700 in this chart is from Matthias's lab, the Craig lab. We have a couple of systems that have ARIScan, the LSM 880 and 980, and there's a STED available through a core facility in our Life Science Institute at UBC. And so, it's kind of fun to take expanded samples to all these different microscopes and see what you can get. And, that became part of this publication. So here you can see some PSD95 labeled and imaged on these different scopes. And I think from the bottom row there, you can see these, we've zoomed in on some of them. And the features in the STED are the smallest, as you might expect. It has the highest resolution. The LSM 700 is clearly the blurriest, but we were kind of hoping that we could make this a bit more quantitative when we were working on this for the paper. So next slide, please, Matthias. So we took some inspiration from this Gao et al. 2019 paper where they were imaging expanded samples using the lattice light sheet microscope. In particular, it was figure 1D, where they explored the performance of different microscopes of interest to them. So maybe go to the next slide. Yeah, so this is the figure from that paper. So they were comparing imaging expanded samples using the lattice light sheet microscope. That's the first column in figure 1D, spinning disk confocal, and an ARIScan confocal. And what they've done there is starting with images like I put on the other side of the slide there, they would calculate the Fourier transform of the image and then try to get a sense of the spatial frequency content of this thing. So these arcs are meant to guide us a little bit and what that means. So higher spatial frequency corresponds to a smaller feature in your sample. So the red arc there at 200 nanometers is kind of something like the limit we are going to have for an optical microscope. And so the brighter this plot is towards that red arc, the more information that you're going to get out of your image at a finer spatial scale. So we thought this might be a good idea to compare the different microscopes that we had for our expanded samples. So we did something like this. Matthias, next slide. So these are from images like you saw before using the 4 different microscopes that we had, the LSM 700, 880, 980, and the STED microscope. We drew circles here that correspond to the spatial frequency and which length scale that is in the sample. And same kind of idea, the brighter the signal is towards those outer rings, the more information content there is at fine spatial scales. So to make this a little easier to get our heads around, we decided to average this around the origin. So to produce the 2D plot on the other side of this slide. And so here we've plotted the power, so the brightness in that in what's on the other side versus the spatial frequency. And so the red line there, that corresponds to the LSM 700. And you can see that it kind of plateaus rather quickly. And so our interpretation there is that this is some kind of spatial frequency invariant noise that takes over, and you're not fully able to exploit everything that you might get out of this sample with such a microscope. The ARIScan scopes, so these are the green and the purple lines, do a bit better. They're still sloping downward as we go to the finer spatial scales. But clearly the STED microscope is doing better than all of these other microscopes with the potential to get information out to quite small features in the sample. So I think a lot of times the attraction of expansion microscopy is that you could potentially turn any microscope into a super resolution microscope by physically magnifying your sample. But it does matter in the end what microscope you use, and we just wanted to get that across in this paper. So if you really, really, really want to be greedy and you have access to a STED, use it. You'll see interesting things. So I think that's all I wanted to say, Matthias.
Sure. And so, yeah, maybe to just emphasize, ultimately it will be also this kind of plot could be a bit useful in the sense of like depending on what resolution you want to reach at the end and how bright is your signal. Remember that I was telling you about this dilution of the signal. You may not need to even expand your tissue completely. If you have a good imaging system accessible, you may just expand it partially to like maybe 1.5 or 2x only, and then using a better of this imaging system in order to get more signal, but without diluting too much the fluorescent signal of your protein of interests. So maybe just to wrap up a little bit what we talked about. So using this map procedure, we can have an improvement into the visualization and quantification of synaptic protein, especially keeping the spatial distribution of these proteins in tissue. And it's mainly due to a gain in antigen accessibility more than just the physical expansion of the tissue. And so this procedure works in our hand for more than 40 different synaptic proteins of the different synaptic compartment. And then ultimately you can boost the ability of your imaging system using MAP to resolve and visualize like fine structure detail. And so it's really important, as I was saying, to consider what you want to achieve, what kind of resolution you want to achieve. And I think maybe what we can also add on top of this, on top of the resolution, it's also maybe the speed at which you want to acquire, because as expansion microscopy says, you're making your tissue physically bigger. So if you want to image the same amount of biological tissue, of biological sample, you will need to image larger area. And so depending on your imaging system, it means like spending much more time doing imaging. So that's another thing to consider, is maybe using systems that are a bit faster to acquire while keeping good resolution. That's another thing you need to consider while using this. So on this, I would like to thank Jeff first to help me doing this presentation, as well as every one of you who came to attend, and my lab, especially Anne-Marie Craig and Mark Zembrowski, my two supervisors, as well as Dr. Peng Zhang. He is now a professor at Case Western University, and he really helped me a lot, starting with the expansion microscopy, the magnified analysis of the proteome, and he's now using MAP in his new lab, trying to look at I think it's this glycosylation in the synaptic, in specific synaptic proteins, while using expansion microscopy and various imaging systems. So I would recommend you to look also into his work if you're interested into this. So on this, we can open the discussion, and thank you for your attendance.
Wonderful. Thank you. Thank you both so much. Are there any questions? You can raise your hand, you can just unmute yourself, you can add them to the chat. Please, anyone take the floor if you have a question.
Yeah, I go first. I'm not interrupting anyone. Hi, thanks for the talk. It was really, really great. So a long time ago, we had played around with this expansion instead, and we got some interesting results. It's really nice. But the one challenge we really had was that since everything gets bigger, it also gets thicker. So the working distance limitations were huge and were really challenging. So we did exactly what Matthias and Jeff described, where we did this partial expansion, like to still be able to keep it in focus. How did you guys manage this very thicker sample and you can't reach very far with the objective anymore?
Yes, so that's another consideration. I think it really depends on what you are looking at. So, for example, right now, what I'm studying specifically is FFP tissue, so formalin. So it's paraffin embedded and formalin fixed human brain tissue. And so what I'm doing is when I'm doing this expansion microscopy, first I'm making sure that when I'm going to mount the final image, the final tissue, the tissue side that I want to image is very close to the cover slip or directly lying on the cover slip. So I'm reducing the distance that there will be between the objective and the rest of the tissue. But then this is really about trying your imaging system and making sure that your imaging system can go as deep as you would like it to go, right? And so, for example, right now, one of the systems I'm using is the data slide sheet that allows me to go, in my specific case, deep enough to cover the entire tissue that I want to look at. But that's definitely a challenge. If what you want to look at is, let's say, if we stay in the brain, you really want to look at the connectivity of, like, let's say, like the dendrites or the axon that projects from one region to another region, and you would like to trace this entire axon, this entire dendrite. This is thing that we had challenged to do. We tried to do it in the hippocampus, especially like tracing all of these dendrites in the CA region. You can make your tissue thicker, but like ultimately using like standard confocal, you are ultimately going to be limited. Or you can use objective that has maybe a bit less resolution, like not using a 40x with like NA of like 1.4 or 1.2, but maybe like taking a lower NA, having like a bit more depth, being able to go a bit deeper. But yeah, ultimately it's like, I would say really like trying out what your imaging system can do, because you won't be able to adapt physically what your imaging system can do with the tissue that you get at the end. And so yeah, doing this partial expansion might help. But then if your question is really about, so the same, looking at like the projection of specific neuron to another region, maybe this specific technique might not be the best suited. In our center, what people are doing also are clearing with light sheet imaging. And for example, when you want to really look into high distances, that would be what people would do in our center. If Jeff, you want to add anything?
I just wanted to ask specifically for when you did the STED on on these map samples, what kind of objectives were you using? But that's a really interested thought process. That's exactly how you would have to design these experiments, because yeah, things get really big really fast.
Yeah, so for specifically the STED, what happened is, so I didn't do it myself. It was another person who did it. I think, maybe you can correct me if I'm wrong. I think the STED that we have was having was it 86x objective? And so, yes, it was really not deep. So it was just about when we did this experiment with the STED, it was only to see how much resolution we can get at the end. So we didn't go very deep. We really went like at the border of the tissue, and yeah, we couldn't go very deep. Another thing that happened with the STED, because just about the technology of the STED, I don't know at that time. I don't know what's the current state of the technology, because you are using these two beams, like one to extend the signal and one to excite the signal. As I was saying, that your signal is already very diluted. One of the issues that we had was that we were having a lot of bleaching very fast. And so if you want to reconstruct something in 3D, you might do it over a couple of synapses, but it's going to be very hard to like, I don't know, have like a full region and trying to like do this over a full region, because you are going to extinguish a lot of signal, you are going to bleach a lot of signal, specifically with that system at that time. I don't know currently if like there is like some more improvement in to the current step technology.
Thomas. You're muted.
So thank you again. Thank you, Ethan, and thank you, Mattia, for that great talk. Got me really excited. Just a question down the same route as Andrian's question. When you expand your sample, your tissue, how thick is it in the end? And then as an extension of that question, if you know, though, I have an extension to the question is, on your more classical, like your confocal microscopes and the STED that you used, did you use any adaptive optics with it since it's getting thick or not? Probably, I assume not, because that's relatively new in commercial systems, but would be interesting to know.
So for oh, sorry. Yeah. So first of all, your first question was about how thick is the tissue?
How thick is it in the end?
So for the tissue that we were doing section of 170 microns. So ultimately, when they are fully expanded, they are going to reach not the 4 times expansion. The 4 times expansion is on the axial, so no, on the X and Y, but not on the axial. It would be more about yes, like 450 to 500 micrometer.
Yeah.
It's not
And
going
then the
to be
question, because of that, did you use any adaptive optics or just
no, we didn't use any adaptive optics.
Yeah.
We just use, so what the kind of
That would
optics
be
we are
great.
using, yeah, the kind of optics that we are using are either air optics or the one that are like water immersion, because your refractive index is going to be the same as the one of water. So you need to have a water objective to do the imaging. So that's, but we are just using like standard imaging setting, right?
Okay.
So
thank you.
I'll just add one thing there, which is the point that Matthias made about the transparency, I think is quite important for that. So the sample is so transparent, we even have trouble seeing it when we put it on a dish and put it under the microscope. So initially, when we were trying this on our Leica confocal, we would use their, what is it called, spiral scan in the navigator mode, and just look at DAPI and try to get enough tissue imaged there that we could find our way around. And so I remember in the earlier days, before we had this procedure so well worked out and we were playing around with even thicker chunks of tissue that were expanded, and using our 25x objective on that microscope that has, I think it's 2 or 3 millimeters of working distance, and you could just image all the way through this chunk of expanded tissue. So the limitation in getting the signal out is about geometry, more so than what's happening with the light, in my mind.
Thank you.
Yeah.
James.
Hi, thanks, guys. So this is more a question for Jeff. Before you mute yourself too quickly there. So in comparing the different resolutions in the Fourier domain, have you heard of this technique, Fourier ring correlation, FRC? This is something we've been playing around with, and it looks, somewhat similar in the Fourier domain. It takes, you have to do two images, two identical images, and it looks at the correlation between the two images. And where the correlation drops into the noise, that's kind of your limit of resolution, but it gives, there's a nice plug-in. So I don't know, this can maybe be a topic for another day, quantifying resolution in STED and confocal microscopy, but we're actually working on a paper comparing all kinds of confocal parameters and seeing how FRC compares. There's already a very nice paper. I can drop it in the chat on this applied to STED specifically. So
Okay. That'll be helpful. Thank you.
yeah, it's I think it's easy. It's just a plugin, and you literally press a button, and it does the correlation and gives you the resolution. So
Yeah, I would really like to try that. Thank you, James.
okay, you're welcome.
I think, yeah, we've been thinking about doing that, like a bit of an extension of that, since we also have the lattice light sheet images of expanded samples here. So yeah, maybe we can try this correlation method with all of these things. Cool. Thank you, James.
That sounds like it could be a topic for a future presentation. So, James, if you are so inclined to come back, we'd be happy to have you talk to us about that method. I think that would be of interest to a lot of folks in this room
Sure.
for sharing the paper. Yeah. I see a question from Andy. What maybe Andy could jump on and clarify, because I don't quite get it.
Yeah.
What are solution in expanded tissues prior to imaging?
Right. My question is basically before imaging, what's the final step you guys did during the sample preparation?
So for the imaging, it depends if you want to have your tissue fully expanded or partially expanded. If you want to have it only partially expanded, regular PBS would work. And then if you want to have it fully expanded, in the original paper, they were mentioning just water, millicube water. But what we realized is that when you use only water, the expansion factor is not very stable. So what we end up doing now is by diluting the PBS a lot. In the paper, we are 0.01x or 0.001x. So it's just very diluted salt. It's almost water, but just having a little bit of salt, a little bit of buffer in the tissue helps to just stabilize the expansion factor to have it a bit more consistent across the samples.
Great, thank you.
Any more questions? Dylan.
I got a question. So just being ignorant to expansion microscopy as well, is any of it temperature sensitive? If you start running a confocal over the area for a bit and it starts warming up the liquid in the area, will you start getting some significant drift? Will the expansion increase or decrease? I don't know. I was curious if you're running into any issues with this.
That's a very good question, actually. So what we are doing is the way I'm mounting the tissue right now is by almost sticking it to the cover slip. So I'm using like polylysine to coat the cover slip, and then I'm going to mount the tissue on it. So because it's like physically attached to the cover slip, we haven't seen an issue with drifting of the tissue, just of the tissue, or chaining and expansion of the tissue. And also, when we tried at the beginning to do the expansion at 37 degrees versus doing the expansion at room temperature, we haven't seen any difference into the expansion factor at the end. But there are, I mean, depending on your imaging system, you would just have the same issues of like thermal drifting or like drifting in general than any other imaging system. And so especially when another thing, because it's much bigger if you do longer image acquisition of very, very big region. That's something that you really need to pay attention, is because you are imaging larger region of interest, then you might have more drifting because it's just physically bigger. So having a system that allows you to keep your sample in focus if you do very large region is very important here.
Thank you.
Additional questions? Okay, so thank you once again, Matthias and Jeffrey. That was excellent and exactly what we were looking for a STED presentation. I think I will open these breakout rooms. Before I do so, I will say that our next meeting is going to be June 12th, same time. So we hope to see you all again there. Watch out for the announcements on social media, Focal Plane, and the emails from Bina. The breakout rooms will have just a few minutes since we're approaching the 12 anyways, but one will have Matthias and Jeffrey there to answer any questions. And another will be on the proposed speakers for the STED super resolution microscopy session at GBI Cross Bina coming up in October. So if you have ideas